Look at the.
I click on now.
Yeah. Good evening, everyone. My name is Allison Foss, and I'm the Executive Director of the Myasthenia Gravis Association. We're so glad that you're joining us tonight for our April virtual monthly meetup. I want to introduce you to a new face that we have tonight on our panel. Katarina Eads has joined our team about a month ago as our newest outreach specialist. So, Katarina, welcome. You will be seeing more of her face as she trains and she becomes our monthly host for our virtual monthly meetups. So thanks, Katarina, for being here. Also want to say hello to Catherine Clemens. She is joining us. Many of you know her, our community program coordinator in St. Louis. So thanks, Catherine, for being here. Just a couple housekeeping items. We're going to use the Q&A for tonight's session. If you have any questions, you can put those in the Q&A. After tonight's session, you will get a survey. We would love your feedback on tonight's presentation and any future presentation ideas you may have for us. Tonight's presentation is the idea of one of our community members. So we are so thrilled to have Colleen Noviello, I hope I said that right, who's a project scientist from the University of California, San Diego, here to talk about the protein structures in MG. So Colleen, I'm going to turn it over to you and you can introduce your team.
Sounds great. Thanks, Allison, and thanks to the Myasthenia Gravis Association for inviting us to talk about our work. So my name is Colleen Noviello, and behind me is Wanwan Lee. He is the lead author on the study I'm going to talk about tonight. He is a postdoc in our lab, so he has his PhD, and then he came to our group about 3 years ago to get more training, and he really did the majority of the work I'm going to talk about and has just been an extraordinary member of our group. Sitting there is Jinfeng Tang. She's our electrophysiologist, which means she measures the tiny currents that I'm going to talk about today. And she's an incredibly valued member of our research team. She's been with us for many years now, and hopefully we'll keep her forever. And I'd like to introduce our newest trainee on our MG team. This is Ilse Schaffner. She's a PhD student starting her second year, or midway through her second year. And so she'll be continuing the work that Wanwan pioneered, supported by myself and Jinfeng. And so I'm going to go ahead and share my presentation. And before I share it, I want to make sure everyone knows that the first slide is just my funding sources, not the people that I'm speaking to tonight. I know that can be a little bit tricky. But we here in San Diego, okay, you can see my application, and great. Does that look all right? All right, thumbs up, great. So we are funded by your tax dollars through the National Institute of Neurological Disorders and Stroke. It's one of the institutes from the National Institutes of Health. So thank you for that. And we're also funded in part the pilot work through the MGFA, not to be confused with the beautiful MGA that we're speaking with tonight. So I wanted to give a little bit of an overview of what we're going to talk about today. And the first thing we're going to talk about is who we are. So we are a basic science team. We are not physicians. We do not see patients, but we study the proteins involved in myasthenia gravis at the molecular and atomic level. And we use a tool called structural biology to do this. And I'll get into a lot of that as we go through the talk. I'm going to give a brief overview of how the nervous system activates skeletal muscle under healthy conditions. And then we're going to talk about the different ways that the myasthenia gravis autoantibodies can attack the healthy neuromuscular junction. I'm going to talk to you about what we've learned in the last couple of years, mostly through Wanwan and Jinfeng's hard work, and what Ilse and Jinfeng and I are hoping to learn going forward. And that, again, reflects on where we're going. We want to start closing those gaps to help inform the physicians and the pharma companies to develop better medicines for y'all. This is a really important context. Again, I just want to remind you guys, we are not physicians. We are not accepting patient samples. I'm sorry if that is disappointing, but we are really dedicated to try and help improve the lives of people with MG. All of the people in this room are working on this, and we care. Okay, so who are we? So on the UCSD side, there's myself, there's the professor Ryan Hibbs, and these three folks in the room, which I'm really excited that they're here. And we collaborate with a group at Yale led by Kevin O'Connor, and he works, most of the work today is performed in collaboration with his former grad student, now postdoc Min Pham, and he is recently joined by Ailish, who is continuing the work on their side. So let's give a brief overview of neuromuscular signaling at the cartoon level, and we're going to keep zooming in, but I wanted to make sure we're all on the same page. So when you have a healthy neuromuscular system and you go to the physician and she taps your knee with a hammer and your foot kicks, that's activating the myototic reflex. So myo means muscle. And you've got this blue sensory neuron going straight to your spinal cord. And it doesn't have to go up to your brain. It just immediately goes back out to your motor efferent axons and your neurons. And your neurons synapse on the muscle, and they release something that causes your muscle to contract. And that's your reflex. And the things that are causing your muscle to contract are neurotransmitters binding to receptors. So if we go back to these little finger-like projections and we look at them even closer, these are these finger-like projections coming down on these muscle fibers. So the muscle fibers are extended, and then when they contract, they overlap. And what triggers that contraction is a neurotransmitter called acetylcholine. Many of you are probably familiar with acetylcholine because you were tested for acetylcholine receptor antibodies. But before you can get to the acetylcholine receptor, you have to release acetylcholine. So you've got these synaptic vesicles filled with this little chemical, acetylcholine, and it crosses the synapse and binds to these receptors sitting at the top. Well, then what happens? These receptors are ion channels. So remember from your high school chemistry, ions are charged atoms, in this case, sodium, going through this little receptor sitting on the membrane. And these ions crossing the receptor create an electrical current. When acetylcholine binds to the receptor, it opens the ion channel. And then what happens is this increase of charges creates a local depolarization. It changes the polarity or the balance of electrons across the membrane. And that triggers voltage-gated channels down here. And those voltage-gated channels actually cause your muscles to contract. Did I get all that right, you two?
Yes.
All right. Let's check in with the
Colleen?
expert.
Colleen,
Yes.
someone is commenting in the chat that they can't see the bottom.
The bottom of the slide.
Yeah,
But not where?
Can you see the bottom of the slide?
I can.
I don't think I'm going to be able to fix that, unfortunately.
Okay.
Maybe they can adjust the resolution or
Do you
something.
think you're into a pointer? Well,
It's not the pointer. They can't see the bottom of the slide.
I know, but in general.
Sure.
easier to.
Okay. They want me to make my arrow options visible, like that.
No, makes your mouse into a laser
Point,
pointer.
laser point. See there.
Okay. Other people are saying they see it fine, so it might just
Okay.
be the size of the screen.
I think it's just something with their resolution. I'm sorry.
Okay,
Okay.
thank you.
No problem. So back to the ion channel and the conductance, and that triggering the voltage gated channels at the bottom of the synaptic cleft, triggering voluntary muscle contraction. So if we look at this even closer, This is a cartoon that Wanwan created, and here are the neurotransmitters being released onto an ion channel, and the arrow represents the flow of ions. Well, this is the real thing. So this is a scanning electron micrograph taken at very high magnification. And on the bottom, there are the muscle cells itself, the tubes that overlap to contract and extend to relax. These are your neurons synapsing. So these little circles are the synaptic vesicles that release the acetylcholine. These are the energy factories of the cell. And probably your eye is drawn to these black squiggly lines. What are these? So this is radio-labeled snake toxin that binds specifically to the acetylcholine receptor. And the radiation from this tag exposes the film really brightly, so it comes off as this weird pattern. But essentially, it's showing you where all of the acetylcholine receptors are found along the neuromuscular junction synapse. Okay. So let's talk about what happens in myasthenia gravis. I think all of you patients could give us a masterclass on this. But from a scientific perspective, it's been studied for decades, and it is the prototypical antibody-mediated autoimmune disease. Well, just because it's been studied for decades doesn't mean we understand it very well, but we understand it better than a lot of other autoantibody-mediated diseases. We know, for example, that the B cells generate autoantibodies targeting skeletal muscle proteins. In addition to the acetylcholine receptor, which is what we work on, other proteins at the neuromuscular junction that can be targeted include MuSK or LGI1. You guys all know that it causes a wide spectrum of muscle weakness. Often it first presents in the ocular muscles, causing that droopy eye or ptosis. It affects your movements. Bulbar symptoms can affect speech and swallowing, and unfortunately, in really severe cases, patients can go into a respiratory crisis. For our lab's work, we specialize in the protein superfamily that includes the acetylcholine receptor. And so, unfortunately, when 85% or more of the patients have these antibodies that target this muscle acetylcholine receptor, well, it's really the most important protein at that junction, I would argue. And so we were really motivated to study this disease because we wanted to help all of these patients. Okay, let's talk about how these antibodies can affect the acetylcholine receptor. So many of you have likely heard of complement, and complement is a natural part of our immune system that the body has evolved to pop bacteria or to kill viruses. The complement sits on the tail end of a normal antibody, and the normal antibody should be binding to, say, bacteria or something else that's pathogenic. But in this case, we've got acetylcholine receptor antibodies, and then the complement sits on the tail end and triggers this cascade, this membrane attack complex, which punches holes in the neuromuscular junction. Another way that the antibodies can affect the acetylcholine receptor is by reaching across and pulling two acetylcholine receptors close enough that they are then triggered to be internalized or removed from the muscle cell surface. And finally, the third major mechanism by which these antibodies attack acetylcholine receptors is thought to be to directly block the acetylcholine from binding in the first place by physically occupying the neurotransmitter binding site. So as you might imagine, and you might be familiar with, pharma companies have developed several ways to target these mechanisms, and perhaps one of the more recent ones that you may have tried are complement inhibitors, which are really promising, but don't necessarily address these other mechanisms by which acetylcholine antibodies can affect the neuromuscular junction. Other therapeutics, which you may have experienced, include pyridostigmine. So pyridostigmine prevents acetylcholine in the synapse from being degraded. So it's trying to increase the total amount of acetylcholine, but if there aren't receptors there to bind it to, it's not necessarily going to help much. Steroids are a general immunosuppressant. These newest ones are the FcRn inhibitors. And where they work are when the antibodies get internalized along with, say, the cross-linked receptors, the antibodies get spit back out and can do their pathology all over again. But the FcRn inhibitors prevent the antibodies from getting released again, so it decreases the total amount of antibodies that are floating around and can cause pathology. Rituximab, which isn't too commonly prescribed for MG, but is around, prevents B cells from being expressed in the body. They're a B cell depletion therapy. And probably one of the more common treatments is thymectomy. And what your thymus does is, in healthy thymuses, it educates your B cells and T cells, really educates your T cells to prevent them from being autoreactive. But in MG patients, often their thymus gets dysregulated, and that increases the rate of autoreactive B cells. So removing the source of the autoreactive B cells helps reduce symptoms in many people. So if we know so much about this disease, and we have been studying it for so long, what are the major questions that remain? I see a pop-up in the chat, Allison. Should we worry about this? Let's see.
I
Move
don't think so, no.
the bottom of the screen. We'll put the Zoom options bottom. Oh, that was probably a healthy tip for from someone, or a useful tip from one of the people to the other people. Okay, so we study the acetylcholine receptor, and we study what's called its gating cycle. So I mentioned this is an ion channel. Well, this channel isn't always open. In the absence of acetylcholine, the channel is closed, and it's what we call the resting state. The acetylcholine is around for only a little bit of time, and it opens the channel and allows ions to go through. You don't want your muscles always to be contracted. If the acetylcholine sticks around, then the desensitized state happens where the acetylcholine is still bound, but the bottom of the channel is closed. So we wanted to ask, how do these patient antibodies interfere with this gating cycle? So our lab specializes in studying the currents of this receptor at the molecular level. And we want to know, how do patient antibodies affect those currents? Because without those currents, you don't get muscle contraction. So in addition to complement activation, internalization, and blocking, we want to understand how these antibodies affect the channel itself. We also wanted to understand, can we map receptor epitopes? So an epitope is, what an epitope means is where an antibody touches the receptor. And there has been some general ideas, but we want to map it at the atomic level, because then we can inform better, are these epitopes conserved across different people? Are there hotspots where many people's antibodies are hitting the same place on the receptor, and we could potentially suggest that as a druggable target? Or are they all over the place? We also want to know, how common is channel antagonism? How frequently is this part of the receptor interfered by antibodies binding to this part? And finally, these antibodies can be multitaskers. The O'Connor lab at Yale has described that one antibody can do all three of these functions. They can block, it can activate complement, and it can internalize. So we want to understand how one antibody can do all of those things. So why haven't these questions been answered before? Well, he wasn't working on it. So we have some truly extraordinary people in our lab, and the technology has gotten to a place where we can use tiny, tiny amounts of protein and understand it at the atomic level. But in order to get tiny, tiny amounts of protein, we have to find a source for it. Historically, the source for muscle type acetylcholine receptors have been from other species. So there's this torpedo fish, which is an electric eel. And what generates the electricity are thousands of copies of a related acetylcholine receptor. But we can't use that because torpedoes fish are very different from humans, and the antibodies in humans don't bind the torpedo fish. We thought about purifying the native acetylcholine receptors from cows, but again, human antibodies don't really cross-react with cow receptors. We didn't want to take native human samples from people's muscles or from cadavers because not only would it be unpleasant for humans to donate their tissue when they're still alive, the quantity of muscle acetylcholine receptor targeted by these antibodies is so tiny, it's not really feasible to study a lot of different patients' antibodies. So what we ended up doing is trying to express DNA pieces from the muscle acetylcholine receptor in cells in a flask. But why has this not been done before? It seems like a really obvious thing for scientists. The reason it hasn't necessarily succeeded before it's been tried is there are 4 different kinds of parts of the acetylcholine receptor, and they assemble as a 5 or a pentamer. So you have alpha subunits, you have beta subunits, you have epsilon subunits, and you have delta subunits. And if you just throw all 4 of those DNA chunks into a cell, they mix and match in different combinations, but there's only one correct combination. And so in order to get enough of this to study patient antibodies, we tried a lot of different things. And eventually Wan came up with a way that actually mimics the way these genes are assembled in our natural DNA. So we had to pair the alpha and delta subunits in one piece of DNA and the beta and epsilon subunits on another piece of DNA. And in the lab, these are just circles of DNA, not whole chromosomes. And we took a bunch of other tricks where we fused them and purified them. And this is a biochemistry trace, and I'm not going to go through all of the nitty-gritty details, but what I hope you can appreciate is that this nice tall peak is frankly a really good indicator that we've got all of the subunits. It's eluting at the correct time in our experiment, and so this was really encouraging. And then there are multiple bands here, and we cut out these bands and did a protein sequencing method to confirm that all 4 of those subunits were present in this beautiful tight peak. Okay, so all 4 subunits are present, but they could still be mix and match. How do we prove that they're actually correctly assembled? That's where we turn to our structural biology method, and it's called cryo-electron microscopy. So what is cryo-electron microscopy? Well, we take our purified protein, and this is just a cartoon of it, not the way it's really shaped, and we put it in ice. And we freeze it really carefully, and we hit it with a huge electron beam. So this beam is 300 kilovolts, and it's focused into a tiny, tiny, tiny central beam, and it hits those protein molecules, and then those projections are collected on a camera that has a shutter rate so fast it can see the little proteins move in the ice as the beam hits it. We sum up those little movies, and we take the computer program takes boxes around each of these different protein molecules. So each one is a separate protein containing all the parts of this particular protein that we want to study. We take those and we average all of the ones that have the same orientation or have the same angle. So if you think about looking at someone straight on, they look different from the side and then from the back. But if you want to get a 3D picture of someone's entire face, you have to look at all the angles. So the more angles you have, the better. Then you can take those and create a 3D representation of all of the angles of this protein. And this is what we call the map. Okay. What we can do with this, and what Wan did with this, is he got these beautiful density maps. So this is the actual protein density map from electron microscopy. And he did it in the different states of the gating cycle without antibodies at first. Because in order to study the receptor with the antibodies, we had to establish what it looked like without the antibodies. So again, the resting, the open, and the desensitized are all part of this receptor's gating cycle. And here's a beautiful measurement of the current through these receptors done by Jinfeng, showing that this is what the electricity looks like when it's going through the receptors. Without acetylcholine, there's no conduction of current. And this part of this trace is what this protein looks like. So you've got this tight spot looking down the top of the protein. Also note you have all of the subunits. You've got 2 alphas, 1 delta, 1 beta, and 1 epsilon, and they're assembled correctly. And this tight little spot is too narrow for the sodium ions to go through. That's what represented by this red spheres. And this is the part of the ion channel that we call the pore. So this moves in the presence of acetylcholine. And Wanwan carefully added acetylcholine and showed where it bound on the receptor. This is our native neurotransmitter. It binds in 2 places. And then you can see at the bottom here, the ion channel pore is a little bit more open. Now it's the desensitized state, so it's not actually conducting electricity, but we can look at these 2 reference structures of the natural gating cycle of the acetylcholine receptor and understand how they change. So I think this is a good point to pause. My mouse pointer is really tiny. Can you possibly make it larger? I will try. If anybody has any scientific questions, let me know. Is this the one you want? Red laser
Yeah.
spotlight?
Maybe not.
That's that.
Maybe that's.
Hey, you increase the,
I don't know how to increase the size. We can do an arrow
Oh, weird.
and then color. I don't know. Oh,
No.
nope, that's not good. Well, while I figure that out, does anybody have any questions on the content? Hopefully that's
Maybe
better.
that's it.
Sorry, that's probably as good as I can get, but let's see. We've got a Q&A. Can the ion channel be constricted in numerous places? What a fun question. Yes. So in fact, the ion channel is constricted here and here. And depending on what drugs and what conformation of the gating cycle, so that's actually really fun. So the resting state gate is up here. So as you imagine, the ions are coming down from the top, they get stuck here first. And then as they go through, the desensitized state gate is down here at the lower part of the channel. So this section is the same part of the protein as this section. That's a fun question. All right. So now let's look at what these receptors look like in the presence of patient antibodies. Okay. How do I get this to go away? Just an X. Okay, cool. So we selected 6 different antibodies from 6 different patients. And we wanted to sample as broad of a scope of mechanisms as possible. And these antibodies were cloned in the O'Connor lab. And the way they got these was through a clinical trial. So for any of you participating in clinical trials, please know that if you sign a research waiver, we may be able to use them someday, and our collaborators will. And it's really meaningful, and your gift goes a very long way and hopefully helps you too. So this is where we got these from. And there we go. I don't know what happened to my laser pointer. So these are all different density maps of different patient antibodies. And the receptor is the center thing of each of these. And these colorful things sticking off or sticking straight up are the different patient antibodies. And what I hope you can appreciate right away is that none of them are the same. Just like MG patients, MG is considered the snowflake disease. Well, part of that is because all y'all's antibodies are binding different parts of the receptor. So the ones that have two of these little arms coming off are binding to those alpha subunits. Some of them are just binding to a delta subunit. Some of them, and we're going to get a little bit deeper into this, are sitting on top of the receptor and almost plugging the top of the receptor. And that's something that had never been seen before. And we also have antibodies that are binding the beta subunit. And so we're going to dig a little bit deeper into these, what we call plugging antibodies, because these antibodies, nobody predicted this mechanism. Nobody had ever seen this mechanism before. When this first showed up, we were all just kind of blown away. There had been some hint that antibodies could bind alpha or delta or interfere with the receptor neurotransmitter site, but nobody had ever guessed that the antibodies would do this. And so let's look a little bit deeper. So this is a cartoon representation of a density map. And this is a loop from the antibody, and this is a loop from the second antibody. And what's really intriguing about this is these are from two different patients, two different antibody genes, but they both do the same thing. They have a conserved mechanism where a loop from the antibody is reaching down inside the receptor's extracellular end. What is it doing there? It's not just physically plugging like a cork in a wine bottle. Remember, these are ion channels. Sodium ions are positively charged. And if we cut the receptor in half and look at the charges of the inside of the receptor, in a healthy patient, these are negatively charged. Well, remember, opposites attract. And so this negative charge is acting to pull in and funnel the receptor. Is it too hot?
Okay, good.
Okay. Pull in the ions for the extracellular domain. Sorry, hold on one second.
It's okay. I'm okay.
Okay. Okay. Pull in the ions, but this antibody is sticking in there and neutralizing the charge. So it's reducing the ability of the receptor to attract those sodium ions into the pore, or into the vestibule rather, and then funnel them down to the ion channel pore. So that's wild. So how do we confirm that? These are structures that generate hypotheses, but we have to show it's actually doing this in cells and not just in purified protein. And that is where Jin Feng comes in. So these are measurements of electricity going through a single channel. She sticks a tiny glass pipette onto a cell and measures these tiny, tiny currents, these picoamps. Everybody's heard of a 20 amp fuse in their house. These picoamps are 10 to the minus 12th amps. They're so small. But when they add up, that's how our muscles contract. So she measured without any antibody around how much current was going through a single channel. So the C means closed, and the O means one channel is open, and the O2 means two channels are open. So when you add acetylcholine, you can see it goes from resting or closed, one channel is open, two channels open. And we measure the height of each one of these channel openings in the absence of antibody or the presence of antibody. And when the antibody is around, the height gets smaller. And we quantify this and see that it's statistically significant. And so we confirmed our hypothesis that these antibodies are in fact decreasing the amount of ions going through the muscle acetylcholine receptor. And that's going to reduce the efficiency by which your muscles can contract, because you need a lot of ions to trigger the voltage gated channels to trigger muscle contraction. The wildest thing that came out of our study is that we found that all of the antibodies we have studied thus far directly inhibit channel activity in slightly different ways, but they all affect the ability of the ion channel to allow those sodium ions to go through. Okay. Another thing we discovered, and again, you're probably not surprised, is that the places where the antibodies bind are all over this receptor. So now we're looking at just the top half of each subunit, color coded. And what we learned was that the ones in red are the spots on the receptor where the antibodies touch. And the main immunogenic region, which was historically thought to be the most important part of the receptor that the antibody touches, was not actually contacted across these 6 patients. Now, that's not saying that we have sampled extensively all the patients, and we are pursuing that, but we really want to communicate to the field that if you're only focusing on the MIR, you're missing a lot of people. And so to reiterate, we learned that yes, the alpha subunits hold more epitopes, but the main immunogenic region is actually not attacked. And we also learned from those plugging antibodies that a surprising place for antibodies to interact is the inside of the top half of the protein. Okay. Before I go into that, does anyone have any questions at all that they wanted to ask? And wait for a moment. Okay, just going fast. Great. So then finally, we wanted to learn if our structures can help predict the broader pathogenic mechanisms. We are channel specialists, but we don't want to ignore the fact that complement activation is a huge part of the pathology of myasthenia gravis. How can we use our structures to inform on how complement might affect these proteins? Well, we looked at how complement binds to these antibodies. And complement likes to sit on a platform of 6 tails of these antibodies, the FC portion. They like those hexamers. Can we predict using our structures how these antibodies binding to the receptor might form that hexameric platform? This is really future work. It's very speculative right now, but what we're hoping to do is start creating enough templates where we could start to predict whether an antibody would efficiently enhance complement activation versus not. Another mechanism that we wanted to examine in a structural perspective is that cross-linking and internalization. Do the antibodies bind in such a way that they could promote the cross-linking and internalization? And I should mention that both of these mechanisms have been validated previously by our collaborators in the Yale O'Connor lab. And so we started to measure these things and see if we can start predicting how more antibodies might behave. So to summarize, we have confirmed that myasthenia gravis acetylcholine receptor antibodies display enormous heterogeneity and diversity in binding. They bind at different angles, they bind in different positions, they bind different subunits, and some of them bind 2 copies versus 1 copy. Really surprising is that all of the antibodies we examined directly antagonized channel activity. They directly interfered with the channel function. They didn't need complement at all. They didn't need to leave from the cell surface. They antagonized the channel. The structures, we hope, can begin to help predict the other immunopathogenic mechanisms of an antibody. And we're trying to encourage people to use our structures as templates to start modeling antibodies from sequences to predict which ones might attack the acetylcholine receptor and perhaps predict which ones might activate complement efficiently. So we also totally understand that a major limitation of acetylcholine receptor therapeutics and broader MG therapies is immunosuppression. We're hoping that our work will help generate a more focused treatment strategy. Besides directly antagonizing the antibodies, 1-1 structures provide templates for drug binding for muscle diseases in the transmembrane domain, in regions not contacted by antibodies, but small molecule drugs now have a high resolution template, and that is critical for pharma to develop better medicine that has less off-target effects. One of the most exciting technologies that's coming around, and it's been pioneered by a group at Columbia named Amy Payne, is this CAART approach. So CAART molecules and therapies target the immune system broadly. But the CAAR is for chimeric autoantigen receptors. And what she's doing is using a chunk of the receptor itself, of the acetylcholine receptor, to bind to the pathologic B cell. So instead of killing all the B cells, she's killing only the ones that are coded to bind the acetylcholine receptor. We've got some questions popping up. So exciting. Any plans to look at samples from MGers in full remission to determine unique binding structure changes? That would be awesome. The tricky thing about the MGers in full remission is that generally speaking, their antibodies aren't at high levels. And so we need antibodies that are at high levels in order to study them. And as far as the way we clone these monoclonal antibodies, presumably depending upon which therapy worked for them to be in remission, it would be more important to look at the B cells. If the B cells are still around, then they've probably gone quiet. Am I aware of any research teams that are taking our discoveries to those of us with MG? So we hope so. So we are giving our work to the biotech companies that work on MG. I don't think I'm allowed to say which names, but we are trying to present our work as useful to them and make them aware of us so they don't only focus on one avenue of therapeutics. Thank you, Valerie. So seronegative, we always get this question. We care about seronegative patients. Approximately 20% of seronegative patients do test positive for acetylcholine receptor antibodies in the clustered cell-based assay. That's work that's done by Kevin O'Connor. And unfortunately, I think for patients that don't have known targets that are truly seronegative, I think that, so I think that essentially you're going to need some like very specialized technique. But what I can tell you is that for 1-1's current and future work, we are looking at small molecule drugs to boost the acetylcholine receptor expression, or acetylcholine receptor function. So if you do not have antibodies against the acetylcholine receptor, then we are trying to work on developing better drugs to help the acetylcholine receptor open, whether or not you have antibodies to it. When you have a very low positive binding acetylcholine receptor, negative blocking, modulating, and striation, a normal RNS and EMG test, normal CT scan of thymus, does that mean you have MG, or could another disease cause low positive? Would immunosuppression still be needed for what I'm being told is a mild case? So I want to remind, I'm not a physician, so I can't give you patient advice. But what I would suggest is that titers don't necessarily correlate to symptoms. And so I would definitely talk with your neurologist and your physician. I think that there are other diseases that have acetylcholine receptor positivity. The thymus expresses the acetylcholine receptor subunits. And so if you have other things going on, you can test positive for the acetylcholine receptor antibodies. But if you have symptoms of myasthenia gravis, then you should absolutely talk to your doctor for the best options for you. I'm so sorry, I don't feel comfortable answering whether or not you would still need immunosuppression. I love all these questions though. Feel free, guys. Thank you for the seronegative love. We do care, I promise. We hear you guys. We know we can't imagine how frustrating it must be, but we definitely care. And then finally, I do want to, I'm going to go ahead and close the chat for just a moment. And I do want to acknowledge the lab and especially the patients. Sincerely, we really appreciate you guys. Again, these are our funding sources. Ryan is here. He is our PI. He's our professor. He creates a really incredible environment for all of us to do our work. Wanwan is over here. He's also behind me. So happy. Ilsa is here. Jinfeng is here. But we have a really vibrant research group, which is supported by your tax dollars. Please continue writing your Congress people, keep telling them our work is important, that you care, lobbied like heck for them. It really will matter. Kevin O'Connor is an amazing collaborator at Yale. He really led us. We've also got collaborators working on MG and other autoimmune diseases at UT Southwestern. And with that, please bring on the questions. Oh, two more? Yeah. Oh, we're excited about these questions. 6 autoimmunes. Oh man, Patricia, yikes. Yeah, sure, that makes sense. Yeah, so more questions, please bring them on. Allison, do you have some questions for us?
That was so fascinating. You guys did a really good job of educating us at a level I think that was nice for us to understand. So,
Sure.
and thank you for taking the time to. I know you all
Oh.
have busy lives and things to get to, so we really appreciate the efforts.
No, it really means a lot. Well, I've talked a lot. What do you guys think? Are you happy to be here?
Yeah, sure.
Got another question.
Oh,
What do you wish you knew and understood to get you to the next level?
Ilsa, what do you wish you knew and understood to get you to the next level?
I have to think about that one.
Which parts of MG do you find really intriguing?
I would say really how B cells and T cells are training each other to target the acetylcholine receptor, and really what goes into the early stages of developing the disease, because we're trying to understand a lot of how the antibodies are targeting your receptor at your neuromuscular junction. But I think there's a lot left to learn about how do your B cells even encounter your receptor at the early stages before you even have symptoms, and what goes wrong in your immune system training where you get these autoreactive B cells that make these antibodies. I think that I would be interested in more
Yeah,
research to learn what goes wrong
yeah, nice.
further up the pipeline,
Yes, exactly.
stopping it earlier. Yeah.
How about you, One?
I think I'm going to just mostly focus on the drug discovery. Yeah. Small molecule drug, but specifically help to rescue the muscle receptor dysfunction. Either in the myasthenia gravis as well as in congenital myasthenia syndromes, which are both because the muscle receptor dysfunction. Yeah, we have some very good initial data, which is under in development now. So I hope later, maybe next, let's say few years, we can have a very powerful, very, specific with zero side effects, let's say, hopefully, yeah, to help these different patients with the muscle weakness. Yeah, this I'm going to do, and my future lab, we're going to focus on that.
One's starting a whole lab focusing on improving muscle receptor function.
Yes.
And partnered with naturopaths versus just biotechs and pharmaceutical. No, we, right now, we're not even officially partnering. We're just presenting our work to interested
Yeah.
people. What additional aspects of this research are you considering pursuing? So, yes, the webinar is recorded. So Ilsa is continuing the work on patient antibodies, and we can't give too many specifics because it's unpublished and we want to protect her career, frankly. But we have a very competitive field. But I think, like she said, we want to understand not just causes, but also like what can I say? The way things vary between patients. We want to look more both within one patient, how diverse is the immune repertoire, and across patients.
Gender or whatever.
Yeah.
Yeah.
Yeah, and like differences between early onset, late onset, the different disease subtypes, seeing if there's differences in your antibody targets. And so really that involves just sampling more patient-derived samples and more of your antibodies and, so just a lot more patients, yeah.
Trying to understand everything we can about all you people. It's not surprisingly, people are very, very complex. Has anyone had a reset CAR T cell therapy that cured MG, or does it just put to remission? Great question. I am not sure. I know about the lupus case where it was completely reset. I think someone has had a reset CAR T cell therapy, but I personally don't know the data on that. I don't know if any of our other people here could answer that. No, no. Sorry, Patricia, we don't know. Okay. Oh, we have 7 more? That was all of them, right? Okay.
Yeah.
Cool. Oh, that's fun. What do I need from the people with MG? Write your Congress people. Remind them that we care about you, and there are many other labs working on MG as well. And generally, basic science does matter. It's not just biomedical. So everybody may or may not know about this crazy protein cloned from a jellyfish called green fluorescent protein. And some science nerd, probably someone very much like my son, swam out in the ocean, found a jellyfish, and saw it glowed green. Well, that little green glowing protein has been fused to thousands of proteins since and leapfrogged the field ahead. It won a Nobel Prize, but we use that as a tool for biomedical research. So even basic science that doesn't have a direct disease impact matters. Scott, I think I met you before, actually. Good to see you, man. We are actively applying for grants with the NIH. We are training people. We're working really hard. There are many patient organizations continue supporting each other. We just need you to be around when we have questions, I guess. So thank you.
And maybe there's more if we scroll down.
Oh, apparently I need someone who teaches me how to use Zoom. Is anyone studying exercise and receptors? Yes, we are not, but I saw a presentation on it at an international conference. And I think the bottom line is exercise helps. There is something called a safety factor. So the immunomodulation, the cross-linking and internalization part, it's thought that that is less of a problem than complement, because when you exercise, you can actually strengthen your body against that. Again, I'm not a physician, but my takeaway from the talk that I saw presented was that when carefully done under supervision of a care provider, exercise can be really beneficial. Allison, I would love, or Catherine or Katerina, I would love for you guys to step in with your opinion on that if you know of anything towards that.
I don't feel qualified, I guess, really to share my opinion. I don't know,
Fair.
Catherine, if you have anything to add.
Not related to specific receptors, exercise and receptors, but we have had other webinars related to exercise and MG and ways of doing exercise safely, but nothing specific between the connection
And
of the two.
that is true. We've done, and we have on our YouTube channel, other webinars about exercise and MG.
Yeah, so
As someone living with MG, I think you just have to do what you're comfortable with.
Yeah, absolutely. But and from the science lab rat perspective, they did show an improvement, but what it is for you personally, obviously you're going to be the experts. Thanks for the question, Colleen. Okay.
Are there any more questions? These were fantastic.
one more. Oh, yeah. Oh, great. Thanks, Mike. Thank you, Allison. Thank you for inviting us here and giving us this chance to talk to the patients and talk to all of you. Talk to the people. You're not
Yes.
just patients, you're people, darn it.
Well, thank you for spending part of your evening with us. We are so glad that you joined us. Just one quick note, our May virtual monthly meetup is going to be the week before, due to Memorial Day. So we'll meet on Monday, May 18th. It does look like you got. Oh, no.
Yeah. Hi, Laura. No problem.
Wonderful. Well, thank you again, everyone, for attending. We are recording, and this will be up on our YouTube channel later this week. So
Sounds
thank
great.
you. Have a good night.
Have a good night, everybody.
Bye.